Hello and welcome to this video. In this video, we will describe the different steps of Krebs cycle, which is also known as citric acid cycle because one of the first intermediate that is formed is citrate or citric acid. Also known as TCA cycle, tricarboxylic acid cycle, because most of the intermediate compounds are 3-carboxyl acid intermediates. Krebs cycle is a cyclic process. All of its enzymes, except for one, they are present in the matrix of the mitochondria. So this cycle occurs in the matrix of mitochondria. First of all, the first step is there's that acetyl-CoA, which is 2-carbon high energy compound that is mainly formed from the glycolysis of glucose into pyruvate, and then pyruvate forms the acetyl-CoA. Even the beta oxidation of fatty acids form acetyl-CoA, and some of the amino acids also, the end product is acetyl-CoA. So this is the central key compound of all metabolic process, namely the carbohydrate, lipids and proteins. So this acetyl-CoA, which is 2-carbon high energy compound, this combines with oxaloacetate, which is 4-carbon compound. A water molecule is added and a coenzyme is removed, and this is converted into citrate or citric acid, which is 6-carbon compound, intermediate compound. And the enzyme that catalyzes this reaction, the condensation reaction, is citrate synthetase. In the second step, the citrate is rearranged and is converted into cis-aconitate, catalyzed by the enzyme aconitase. A water molecule is removed in the presence of iron ions and then rearranged again to form isocitrate. So citrate is converted into isocitrate. Then this isocitrate undergoes dehydrogenation. One hydrogen is removed and this is carried away by the carrier NAD. So NADH, they are formed, which then enters the electron transport chain to form the ATP. So isocitrate is converted into oxalosuccinate, 6-carbon compound, and this reaction is catalyzed by isocitrate dehydrogenase. Oxaloacetate in turn undergoes decarboxylation. One carbon dioxide is removed in the presence of magnesium ions and is converted into alpha ketoglutarate, which is 5-carbon compound. Now this alpha ketoglutarate undergoes multi-step reaction to form succinyl-CoA, a 4-carbon intermediate, and the reaction is oxidative decarboxylation. That is, one hydrogen is removed, carried away by NAD, the hydrogen carrier, which then enters, which forms NADH and enters the ETC. A CoA is added in this and carbon dioxide is removed. So this is decarboxylation, and also since hydrogen is removed, so this is oxidative decarboxylation. And the enzyme which catalyzes this reaction is alpha ketoglutarate dehydrogenase complex. Thereafter, the succinyl-CoA is converted into succinate by the enzyme succinate thiokinase. So this undergoes phosphorylation. A CoA is removed from succinyl-CoA and a GTP combines with GDB, and the GDB combines with phosphate to form GTP. So here the GTP is formed. A water molecule is added in this reaction. So in this way the succinyl-CoA is formed into succinate. In turn, the succinate undergoes dehydrogenation. One hydrogen is removed, catalyzed by the enzyme succinate dehydrogenase complex. This time this hydrogen is carried away by FAD to form FADH. and this FAD enters the ETC to form 2 molecules of ATP per molecule of FADH. Now the succinate forms the fumarate in this reaction, and then fumarate is converted into malate by the enzyme fumarase. So fumarate undergo hydration, that is one water molecule is added up to form the malate. Malate in turn undergoes dehydrogenation. So one hydrogen is removed. This is carried away by NAD. So to form NADH, which then enters the ETC to form ATPs, 3 molecules of ATP per molecule of NADH. And now the oxaloacetate is formed. And oxaloacetate again enters the cyclic process to complete another Krebs cycle in this way. So these are all the steps of Krebs cycle, and the enzyme and for this Krebs cycle are in mitochondrial matrix except for succinate dehydrogenase. Succinate dehydrogenase occurs in the inner member of mitochondria, and the net product of Krebs cycle is per molecule of acetyl CoA 2 carbon dioxide are formed, 1 GTP is formed, 3 molecules of NADH are formed, which then enters the ETC, and 1 molecule of FADH is formed, which then again enters the ETC. But if we see per molecule of glucose, because 1 molecule of glucose forms 2 molecules of acetyl CoA. So if we see the end product of with respect to glucose, then 4 carbon dioxide are produced, 2 GTP are produced, 6 molecules of NADH are produced, and 2 molecules of FADH are produced. So this is all about the Krebs cycle. This Krebs cycle is the third step in the cellular respiration, as the first step is glycolysis, the second step is processing of pyruvate, which is the end product of glycolysis, into acetyl CoA, and the third step is entering of that acetyl CoA into the Krebs cycle to release carbon dioxide and number of hydrogen carrier, which then enters the ETC to form ATP. That's all in this video. Hope you like this video. Kindly share and like this video and subscribe to my channel.